The overall goal of this procedure is to model Zika virus infection in the developing human brain using stem cell-derived cerebral organoids.
This method can help answer key questions in the virology field, such as which cells are capable of being infected and which proteins are involved with the infection pathway.
The main advantages of this technique are that it mimics early human infection, can be utilized in high-throughput assays, and can be combined with focused genetic techniques such as CRISPR.
This method can provide insights into the mechanism of Zika virus infection. It can also be applied to other systems, such as drug screens and virus pseudotyping.
Using regular stem cell maintenance methods, bring cultures to between 50 and 70% confluence. Inspect the cultures using bright field microscopy at 10 to 20x magnification and ensure that the colony has a healthy morphology with no detectable differentiation. Bring the xeno-free stem cell maintenance media to 37 degrees Celsius in a hot water bath and thaw 2 milliliters of enzymatic detachment reagent and a 50 microliter vial of the stock ROCK inhibitor at room temperature. Next, prepare an ultra-low attachment U-bottom 96 well plate, a multi-channel P200 pipette, and a 25 milliliter reagent reservoir. Next, aliquot 45 milliliters of the media into a 50 milliliter conical tube, add 45 microliters of the ROCK inhibitor, and mix in the inhibitor thoroughly by triturating the mixture, followed by 2 to 4 inversions. Vacuum aspirate 2 wells of a 6-well plate containing stem cells, and quickly add 1 milliliter of enzyme-free detachment reagent to each well. Then, incubate the plate at 37 degrees Celsius for 4 minutes. Next, vacuum aspirate the treated wells and add 1 milliliter of enzymatic detachment.